Hello everybody, good afternoon from Budapest. My name is Marcello Scala and I'm the Director of Sales for EMEA for Omixon. First of all, thank you very much for your presence at the Omixon webinar. I'm very pleased to inform you that over 200 people registered for this session. Thank you very, very much for that. Here with me, I have our guest, Professor Derek Middleton, Head of Transplantation Immunology at the Royal Liverpool Hospital, visiting professor at School of Biomedical Science at the University of Ulster, honorary professor for the University of Liverpool. He is also author of 350 scientific publications and the originator and webmaster of the Allele Frequencies Online database. Thank you, Derek, to be here.
Pleasure.
I'm also very glad to have here with us my colleague and friend, Dr. Peter Menches, the Chief Commercial Officer for Omixon, who's going to give us a live demonstration of the Omixon HLA Twin software. So, our agenda for today is Professor Middleton will walk us through how to the Allele Frequencies Net database is planning to support the NGS in HNI. And then Dr. Menches will then give you a live demonstration of the Omixon HLA Twin software. And finally, there will be our session where Derek and of course us will answer your questions, so please wait until the end of the presentation. The question can also be submitted by typing it, and Agi, our IT support manager, will collect them for you. So now let me give the opportunity to Derek to continue.
Hi, good day everyone, wherever you are. It's probably morning, night or evening, I don't know. It's a pleasure for me to be here in Budapest, and hopefully I can give you some interest in what we've been doing and what we hope to do on AlleleFrequencies.net database, AFND for short. I want to take you through, and hopefully a lot of you will know the various facets of the website, and talk to you then about more recent innovations, and finally talk a bit more about the future and where we're going with the data, because obviously NGS is going to produce so much data that we do need somewhere to keep it. So just very briefly, this is just the numbers of populations. There's nearly 1,500 populations of various sorts, mainly HLA and to a lesser degree KIR on the website, and we have this is all available on the website. We have maps of where the populations come from, according to which region they're from, and which immunogenetic region they're from. And this is just the means of searching. People can search by locus, by specific alleles, and they can search by a particular population. They can search by a particular country. The type of study, whether it's from anthropology or from a disease controls in a disease association, typing by ethnicity, and we can actually do it by resolution or sample size. So there's all these various things to find out, and hopefully many of you know already about this. And this is the sort of thing that we get when we do a search. This is just by allele, and we have here the population that's in. By clicking on this population, we can get details of that population. We can also see data according to the phenotype frequency, which is really, in this case, 15% of the population carry this allele, and whereas the allele frequency is 0.076. Our sample size. This link here takes you to the IMGT HLA database, where one can find more about that particular allele. Distribution will show maps, and then the haplotype association will give you any haplotype association we have with that particular allele. The data can also be shown by highest to lowest in frequency, or just taking one population at a time and looking at it, or a population at a time, highest to lowest frequency. And this is just the link to IMGT. This is the link to show you more data about the population. Especially maybe important is the notes, which sometimes define whether or not what that particular investigator could not differentiate. These are the sort of maps we have. Not exactly exciting. We probably should be better maps, and we're always trying to improve things, and we do need to get better maps. And these are the haplotype associations. And when someone, and this is more the old way, but when someone actually went to the website and put in their data, this is the sort of data, the demographics that we needed to do. Just going over this very quickly, and it's obviously all available on the website. Another aspect of it is just the massive increase in alleles, as we all know, and I've stopped making these slides. Steve Morris, please forgive me if you're there, at 2012, because it's just the same, it just keeps going up and increasing. But it does bring to us a lot of trouble. So one of the things that we tried to do within the AFND was to keep a record of rare alleles, and that didn't necessarily, we didn't need the frequency of it, because many routine labs may come across an allele and without knowing the frequency of it, but we wanted to try and have that. So we did collect data from IMGT of second sightings of the allele, from NMDP, from other labs, mainly through the international workshops. And so if one then was to click on an allele and it was only had been confirmed or seen once, then we would get details of that allele submission, and especially if it was available a haplotype, which may be of use for other people who may come across and query the allele. Problem is that when people see the allele for the second time, they don't normally report it because, to be honest and cynical, they won't get a publication. So it's a question of trying to collect and trying to find out this, and this is going to even be more important in the NGS era as people see these alleles. So we need to find out just how do they exist. When we did this, especially through the international workshops, it was really, for most loci, about a third of the alleles were frequent, a third were rare, and we counted rare as 1 to 4 occasions that had been reported, and then very rare was just on 1 occasion. Of course, this again is only being reported. They may well have it there and people haven't told us, so we do rely on our colleagues to tell us these things. And this is something that Fabiel produced, whereby people could run this log algorithm with their data to show whether or not the quickly to report these alleles. And of course we wanted to try and tie up with the common and well documented alleles, which has been done mainly followed by Steve Malak, but is really only for those USA, and what we would like to do is just to have rare alleles according to geographical regions, and that was an aim which so far hasn't been fulfilled, but it's one of the things that we'd like to do. Of course, we're not just HLA, and we do the same thing for KIR. We have the phenotype frequency and various things there. We have maps possibly better for the KIR of just showing more of a heat map to show the frequency. And one of the things we did with the KIR was to have the genotypes, and it's nice to know that this genotype number seems now to be used quite a lot. It's not an official number, but it's a number that people usually use if they're reporting the genotypes. So there was a hint there of being able to collect raw genotype data, and according to which populations it occurred in, etc. And I make this point here because it's one of the difficulties we have. People have rightly criticised AFND as regards some of the quality of the data, and a lot of the data has come from peer-published papers. And this is just a point that within the genotypes found within the populations, we have one population, for example, that has 12 genotypes that have not been found in another population, and 2 populations that have 11. Obviously there's something wrong here. It's difficult to know what it is, but it does bring the problem of quality and keeping proper records and proper genotypes and how people have derived their particular genotype. We also have KIR and disease database, whereby people can look up by disease or by KIR gene or by country or whatever and see that, and that again can be supplemented by the frequency of the KIR gene. The reason KIR and not HLA is because we thought we could get in early enough within KIR to collect KIR and disease studies, whereas HLA and disease studies have been going for, well, now 50 years, and very difficult to keep a record. Because as well as those published, we would like to get those not published, because it's very easy, or not maybe easy, but people do publish disease associations when they're positive, but there's an awful lot of negative associations that are never, where there's no association, that are never published. And so the community are not well informed, and that's what we were trying to derive. And so come now more to the last possibly 6 months, 9 months. With the data we have on the website, we decided to try and differentiate it by quality, and we come up with just the gold, the silver, and the bronze standard. And really, the gold was whereby the alleles summed to 1 plus or minus a small fraction for summation, whereby all of the allele names were 2 fields, and the size of the population was greater than 50. The silver, bit similar, the allele frequency summed to 1, but it could be a mixture of 1 and 2 fields, and the sample size could be any, and basically the bronze were anything. So what we're moving towards is this, people being able to use these gold standard populations as being more relevant. You may say, well, why does the allele frequencies not sum up to 1? There must be a mistake, but on certain occasions, a lot of the data we've taken has been from peer publications. And when that happens, sometimes the publication doesn't carry alleles right down to the one. They don't put down the small frequency alleles, and sometimes they just say other alleles and what the frequency of the others are. So that was one way we were moving that, and that's just I hope you can see that, but it gives us an idea of where we haven't got data sets from throughout the world. If there was someone listening from one of these exotic places, maybe they could tell us that. Those with silver here, and then we had the gold according to the depth the color would be related to the numbers. And here we just have this association of where the different loci and what the frequencies were of the different loci in the different geographical regions. There will be a publication I'll refer to later coming out for those where I'm going too quick to show that. And this is just a brief survey of the data that we have for these gold standard. You can see that the majority of populations are from anthropology studies, and this is the number of individuals, but that the bone marrow registries, for example, will cover many, many people per registry, and that the methods that we used according to this, the sequence in the SSOP and the other, and hopefully the future will be next generation sequencing, and perhaps then we then say that a population, and these populations that are typed by next generation sequencing, they maybe could be platinum. So we can always change the criteria to do that, and this just shows the sample size of each of the databases. This is a reasonably new innovation here, which I particularly like. I should say that I like because I don't do any of the informatics. I don't have one idea how to do it. I just hopefully have some ideas, and we're always looking for people to come back and say, well, your website doesn't do this or doesn't do that, and looking for suggestions from others. But this is whereby if you can, it's an active on the website, and you can put an allele, particular allele in this case 1501 DR, and it tells you the number of gold populations that have been typed in the various geographical regions. These geographical regions are those Steve Mack and colleagues came up with in the international workshops, and then tells you the number of populations where this allele is found and the total number of alleles or number of times it's found. And then you can get a summary within Europe that the allele has been found in all these populations, and these numbers relate to the number that is on AFND. So that in itself might be useful for others to know. And then we talk about standardization reporting of next generation, and I suppose that we really should be talking about, it doesn't necessarily have to be next generation, but it can be any method. And a lot of this thoughts and thinking has come from Steve Mack, and you know, the fact that we do focus on names often, and we lose a lot of primary data, that we don't know information is about introns and exons that have been interrogated, and that the database tends to be that what happens is that people do their typing, they probably have ambiguous data, and then it becomes unambiguous from the point of view of reporting frequencies, and that this you know that an HLA genotype represents a unique diploid sequence today, but that may not be the so again when IMGT HLA comes out with a new version of alleles. So we get this fixation in time of the genotyping results, and you cannot actually refer. So to always remember that Steve Mack saying, excuse me, Steve, if you keep referring to yourself, but in the informatics side of things, you are one of the big leaders, but saying that, you know, people might take a half a page to define how they isolated DNA in their study, when we all know how to isolate DNA, but there's no mention of how they got from their primary raw data to having the frequency data, and it could be various means that they've done that by. So one of the things that we've been doing recently, and this is just to try and entice people to give us genotype raw data because it's something that has been missing from AFND, and that is to tie up with Human Immunology and Steve Mack being the current editor of that journal, to have short population reports on the various immunogenetic genes, but it's particularly HLA. To have the demographic information, to have the typing methods, the alleles able to be detected, and to collect the genotype data. To have that ambiguous, warts and all, to have it sitting there, and then to collect the data after ambiguity resolution, if that's been possible, and how it's been resolved. There are a number of, we all know labs that have various ways of resolving their ambiguities, some of which don't stand up to scientific scrutiny, and there's various checks to be done. So this was to try and get people to give us raw data, and in view of the fact that out of that they could get a publication. And we have ways of checking the data automatically for correct nomenclature, whether the ambiguities are resolved, the frequencies reported, that they're consistent with the data. So we're asking people to report their frequencies, but we have ways of making sure that it adds up, and that this will, any problems will immediately be highlighted. And then once it's validated, be sent. And this is sort of thing, curiously, that this allele A3277 is an error, and was an error when this data, we used this data as an example, but would no longer be an error because it now is an allele, but it wasn't at that time. And then we have another allele that doesn't exist. So that highlights it and brings it back to the submitter. And these are the various ways of downloading the data. There are on the website now videos on YouTube to show you just in 3 different sections to show how the data is submitted. And therefore, if there's something wrong, with the various aspects of it, you get a tick if it's okay, and then if there's something wrong, that you get that there's a red mark, an X. In this particular case, the raw data and the frequencies do not match. And then when everything's okay, you get full ticks, you're on board, and you do it. Well, what we've been doing is to try and collect the ambiguous data as such. We would have, for example, 2 lines whereby if there was 2 different choices for the type, that we would put each in a separate line. As you can see in this case, the B locus is not giving the problem. There's no ambiguities about, but the A locus can be either the 010103 and 20104, or 010101 with a 2020118. That's so. This goes further, and this is where it becomes a little bit further away from me, and I'm not so directly involved, but there are many people out there involved, and this was a report in Tissue Antigens a couple of years ago about genotype-less strings. And it just reiterates the problem of ambiguities, and it's not possible to have it that we can resolve always, all the time. So historically, people have used these ambiguities and represent them in different ways, but that then means that we've lost information. So what we're doing with GL strings is to represent the ambiguities in a genotype in a machine readable way. So you've got the allele 1 plus allele 2 or allele 3 and 4, and that you can then accommodate that data for multiple loci from multiple sources. And this is the sort of thing that you get with a type. Oh, goes back to, where's the back? Okay, sorry. I think there was some other movement in that that's got lost. But anyway, there's 5 delimiters to show that. We've got the B alleles as not being unambiguous, and we've got various alleles here. We've got a combination of, that could be that allele or that allele, and it can be those 2 alleles or that allele plus those 2 alleles, and they're represented by these 5 delimiters. So taking that further, the people who are directly involved in this have been using machines to generate and to parse these strings and to collect all the data. And you don't need to be able to read it as long as the machine can do it. So the idea is that somewhere along the line you've got user 1, he puts his data in a GL string, gets it back, but user 2, once he knows the code for that, can get that string and bring it back. So these, and again, some of these slides are coming from Steve Mack, who's more to the fore in this with his colleagues in some of the bone marrow registries. And it's an open source. Anyone can start their own service. It doesn't cost anything. The main thing is it's updatable, that each time there's a new release that can be run on that to analyze, and that, you know, will last forever, long after we're all gone, if it's so desired. And then this brings us to the consortium, that one of the people on it here is Tim Haig from Omixon, but I'm not involved in it. But this is just this consortium of HLA people who have come up with this minimum information for reporting immunogenomic NGS genotyping varying. So it's just a way of keeping this data. So the whole drive is to actually keep raw data, whether you're doing it through allele frequencies and populations and through short population reports, or in cloud type of things that we can then keep forever because we're doing it. And therefore you can actually then take data from the present, compare it with the past, compare it with the future. You can compare data on different platforms from different providers, from different analysis systems, and you can even try and compare data using different, whether it be NGS, SSO, SSP, or whatever. And then when we have all of this worked out perfectly, and there's a big cloud up there containing all this data, I've always believed that the allele name itself, how important is it? It's not really, to me, it's important, and we need that for the nomenclature to know what we're talking about. But at the end of the day, if someone says 0206 is associated with a disease, it's not 0206, there's something in 0206. So this is just a very simple algorithm we had and have had for many years on AFND, which actually converted the frequency at 2 fields to amino acids, and then you could work out what the amino acid was and what the frequency of the amino acid was in that particular disease population or even normal population. One of the other things we've worked on a lot is epitope frequencies from data, and we usually find, well, for example, in the England Northwest, which I belong to now, there's usually one epitope at each of the position which represents the majority, 99% of in that population. So my point is that of trying to actually use the data that we are collecting and have it all there, and then to go back to what you might call functional polymorphism or whatever to know what's really happening. And these are listed, if we do this, we can do the same thing as before, the epitope position according to the population and the frequency of it. And again, it's just a question of drawing these maps. And of course we can do heat maps, we can do trees with these, according, this is the epitopes along the bottom, this is the trees and things like that. This is just a map really to come to the finish of just how many times people have used the website. I mean, we're getting about 300 hits a day, so it's quite popular, but this is just the density map of throughout the world. As you can see, it's mostly colleagues in the States that have used it, but there's also strong use in Europe, in Brazil, in Canada, and down through China and Asia, India, Australia. So it's pretty worldwide that they're being used. And this is just a couple of references that people want. This was the original letter editorial that went from Steve Mack, went out in Human Immunology as regards the short population reports and gives you more details. It's a fixed format of what we want as regards frequencies of demographics. So that the map, one of the things that I had this idea of doing this for many years, because I get a bit tired of reading population discussions that say the allele is in 10% in our population and 9% in adjoining populations and 6% in that way. It doesn't really say anything. All we need is the actual data. So this is a reference to the paper that describes the gold, silver, and what we're doing with the allele frequencies, and more, which will be in Human Immunology, which is in press. And then there is a volume that I'm sure many of you know in Human Immunology last December, which is on the usage of NGS and covers not only the practicalities of the method, but also covers a lot of the informatics side of it. And if you haven't, I would advise you, if you can, to be able to read that. So it only remains for me now to thank the people who've been involved in the website back in Liverpool. There's a guy called Eduardo Dos Santos who was there for a year on a sabbatical, Tony McCabe who's been there for a couple of years now, Andy Jones who's the head of that informatics, and James Jones who is someone who works in the HLA lab in Liverpool but has a meaning towards informatics and is very helpful. Can't thank enough Fabiel Gonzalez Galarza, just Fabiel, we all know him as, who came to Belfast many, many years ago, it seems now, to do a PhD, followed Liverpool, finished his PhD, and is now back in Mexico because he has designed the whole website from the past, and we still keep in contact. Steve Mack, who has been very helpful and encouraging in doing the things and trying to collect that data, and of course Omixon for their sponsorship of the website, for inviting me here, and for all of the other companies who through the years have sponsored the website. So thank you very much for your time and listening, and I hope it's been a quick run through, and as I say, as we've said, and Omixon has said, to take questions, but also, as I can't emphasize enough, but please let us know what we can do to make the website better for you, and we will do our damnedest if we can do that. So thank you very much.
Thank you, Derek. I'm sure that there will be a question at the end of the presentation. Now I think we move to the next item on the agenda, so to the live demonstration of the HLA Omixon twin done by Dr. Menches.
Thanks very much, Marcello, and thanks very much, Derek. It's a privilege to have you here in Budapest with all of us. Good afternoon, everyone in the European time zone. Good morning to everyone on the other side of the world. This demonstration is a little bit more of an advanced demonstration compared to what we would usually give, but I would like to focus specifically on rare alleles in light of Derek's talk about allele frequencies. So for those of you seeing data in HLA Twin for the first time, let me just quickly provide you with an overview of what you're actually looking at. So when you first load HLA Twin, you will see a bunch of your sample data listed here directly in the file system. And what to run any analysis in HLA Twin, you can immediately click on the Analyze button here to start the analysis for a single sample. If you do that, of course, it will start the analysis, but I've already analyzed these, so I'm going to click Cancel. If you wanted to analyze multiple samples simultaneously, you can select the first one, select the last one, and to analyze all at the same time, you can click on the Holotype HLA typing button, and that will initiate analyses for all of your samples simultaneously. Now, in a full webinar on HLA Twin, we would spend more time orientating you in the software. But what we really want to do is show you how the holotype HLA can accurately determine a bunch of rare genotypes. So we're going to view the results that we already have. So to do that, I can actually click on the Show Analyses button, and what I'm going to do is I'm going to hide the samples. So now the data files that you can see have information about the analysis, that we have a number of green lights, yellow lights, and red lights, which are our traffic light system that's associated with the quality of the data for the entire sample. And rare alleles, by definition, are kind of hard to find. But when you do have one, you want to be sure that you pick it up. This is an important part of what we want to show today. The examples that I have here have all been confirmed by alternative technologies from our early access program data. And we have a mixture of 5 locus and 7 locus holotype data, which is from our older kits, because we also now have 11 locus data. And so you may see some in the genotype frequency or in the genotype table that don't show all of the alleles. But to view your samples, I'm going to actually just view everything at the same time. I can click on the View Results button, and for each sample, here you can see the sample name. We have allele 1 and allele 2. We have an HLA-A call, B call, C call, etc. across the row. Now, in this first example, we have, it's actually very easy to see that we've got a rare allele, as noted by the exclamation mark here. So it's very simple. We've got an HLA-A 3148. The question, of course, you should ask yourself is, can I trust this rare allele call? If it's rare, we weren't really expecting it, but can I trust it? In this case, we refer to our traffic light system, in which we have both, we've got 2 green lights. The first of the 2 green lights tells us that the 2 orthogonal algorithms in Twin have both reported the same allele type. And the second traffic light tells us that all of the quality metrics have passed when determining this allele. Now, to have a look at those quality metrics in more detail, we'll just do this by way of reminder. It's important to remember that we have a number of quality metrics designed to give you the maximum confidence in the results that you've obtained. So Omixon HLA Twin reports 18 different quality metrics. You can see a number of them here on the left-hand side: fragment size, read length, cross mapping. And if everything works well, you can see that everything is green. In fact, this lighter green color here for the info light, everything has worked very, very well as well. And this is a really fantastic data. But what we're interested in here is specifically for HLA-A, where we have seen the rare allele. Can we trust this data? Well, right the way through to looking at exon minimum coverage depth and allele imbalance, we have got green lights across the board. If there had been a yellow or a red light, then this system of 18 different quality metrics will allow you to investigate more thoroughly the cause behind the warning. Now, if you're interested in knowing more about all of the different quality metrics and how to do detailed data interpretation, which isn't the topic for today's webinar, you can always email us at support@omixon.com and arrange a webinar with one of our software experts to get more details about that and get a thorough walkthrough of all of the areas of Twin. But what I'd like to show you here today is another tool for investigative purposes in this situation, and that is our genome browser. So because the allele that we're interested in investigating here, the rare allele, is the second of the two that's been assigned. I'm going to click to browse just that allele on its own. And what you can see is that in the data generated by HLA Twin, where it's built a whole gene consensus sequence directly from the data, we've got even and very deep coverage here. You can see the coverage depth is around 140. If you focus down in the bottom left, this area here tells you what the coverage is at any given point. And so you can see we've got really robust coverage across the entire length of the gene. You can see from the phasing track up the top that we have a fully phased consensus sequence across the entire length of the gene from UTR to UTR. And in fact, because there are no colored bars in this gray track here, which is a consensus track in the top and it's a reference track in the bottom, because there are no colored areas, that means there are no differences between the consensus sequence that I've constructed from the sample data and the exons that are defined in the database. So looking here at the second coverage plot, the reason there are gaps is because that's how much has been defined in the database. So we're taking the whole consensus sequence data and we're aligning it to what's available in IMGT HLA database. And of course, if only the exons are defined, we can only display these reads at the positions for exon 2, exon 3, and exon 4. But as you can see, there are no differences between the 3148 in the database and our sample. So from here we can hop back a couple of screens to the original data and have a look at some of the other alleles that we've called. We don't need to spend too much time on them, but here we've uncovered another rare allele. This time it's HLA B15135, again flagged very easily determinable by the exclamation mark sign. In our third sample, we have the another B15, but the B15220. The important thing to notice in all three of these cases so far, and indeed all of the examples that I have here for you, is that we have an unambiguous type with green or green and yellow traffic lights. And that means that this allele has been unambiguously assigned and you can trust the result that you've got. Now, let's have a look at HLA-C. Once again, we've detected a C04108. We've detected a C0635, another C0635, and over among the DRBs, we've got a DRB416, and this is one where it hasn't resolved the ambiguity entirely. Exon 1 of DRB1 is not covered by our primers. It's not a clinically relevant distinction, but we do have the 1210, which is a rare allele, and the 120101, both are equally likely in this situation. So one of the next things that you would do in this, once you have all of this data, is you can select all of your samples simultaneously and ask the software to assign all of the best matches or assign all of the unambiguous matches, assign the concordance, and once all of the allele calls have been assigned, you can then use the approval functions so that everything can be, everything is approved by the time you use it in a clinical report. So I'm going to choose to assign the best matches. HLA Twin will prompt me to, am I sure? In this case, yes, I am. And then I also, for any given for any given sample, I have the ability to make a comment on that sample and then approve the result. And the approval function can also be restricted by user type, so you can have the technician might assign the functions as I've just done, but then the supervisor may be required to approve the result. And so you can distribute those functions across members of your lab. So in this case, I'm going to choose to approve the result because I am my own super user, and that result is now approved. You can see the status has been changed to approved in this column here right at the start. Now, when you've gone through that process for all of your data, the last major feature that I'd like to show you today is how you can export that data from HLA Twin. So we go back to the home screen. You can see because this sample here has been approved, it shows up as green. But regardless of their approval status, I'm actually going to select all of them and choose to export the results. So we're going to export everything. The first thing you need to do is define an output folder. I'm just going to set it to the desktop. That makes it nice and easy to find it, and then click OK. And then of the export options, HLA Twin actually gives you two options. There we go. So we've got two options. HML is actually relevant to some of the things Derek was talking about before. HML captures the GL strings so that those can be reused. It's an open format described by the NMDP and implemented in our software for that purpose. But because it does capture the GL strings, you can include those as well. But what I'd like to point out today is the PDF export function. And you can choose from among the quality metrics, the various data statistics, and other things, which of these you would like included in your report. Of course, the most comprehensive report includes all of them, but it is up to you when adhering with the laboratory standards and the accreditation standards to ensure that you are including all of the details necessary. You can choose to export all alleles or just those that have been assigned if there's anything that is left unassigned. And when you're ready to go, you can click Finish. And the exporting process will begin. You can see I've got one more task running here, and if I go and show the desktop, you can see that I've got some files that are being written here. And for example, I can view the file, the genotyping result, and all of the information for each genotyping result on a per sample basis is available for you to submit as part of your clinical reporting. So that ends the demonstration component for HLA Twin. I'm going to hopefully find, do we need to pop back to the presentation? Okay, so we'll now be taking questions. Thank you very much for those of you who've submitted some text questions. Agi, are you ready to ask those questions of myself and Derek?
Yes, absolutely. So actually, I think you might just want to stay because there was an action regarding the data set that we've been looking at. Which is how would you investigate the B allele with half red, half yellow, second stop and go live? Would you trust that call?
So
I'm not sure, maybe it makes sense to show that data again.
Let's see if we can do that live here, view the results. So, so it's not one of the novel alleles. But yes, so there is a half red, half yellow, which the first thing that we should do is go and look at the sample details. So to do that, we can see that on HLA-B we've got an exon spot noise ratio that is a little higher than we were anticipating. This is actually a small anomaly in the sense that we have since altered the thresholds, and you would not actually this was the newest of the QC metrics that we have. And in testing, we have shifted the threshold, and you would not see a red and yellow light for this sample unless it had more than 20%. So, in this particular example happens to be a bad one to show you how you would go further, because this is a sample that you can definitely trust.
I guess in general it just makes sense to state that when you see something red and yellow, then you will want to have a look at the quality metrics.
That's right. So you will, whenever you see any yellow or red and yellow, our guidance is to inspect and investigate, and so you click on the quality metrics to go and determine the severeness of the issue.
Okay. Yeah, actually there was one more question that was related to the traffic light, so I guess it makes sense to discuss it now.
Sure.
Which is what do the mixed lights mean, and how many levels of traffic lights are there?
So that is a great question. There are 3 colors, obviously, red, yellow, and green. But there are also some half traffic lights. A green yellow is in between the green and the yellow, and so it's intermediate in its interpretation. So it stands for an info light, and the way that you interpret any light except for a red light, so is as passed, but you may have to do a little bit more work. So a green light means that it's passed with no work from the technician. A green and yellow light is the info light, which means there's some information there. Perhaps you've lost phase, but you've still got an unambiguous genotyping. It essentially means you have nothing to worry about, and you can accept the typing without any extra effort. If you have a yellow or a yellow-red, we advise that you inspect or investigate, and those are broadly similar. And what we would advise is that you check the quality metrics first, see which of those quality metrics have been given the yellow or the yellow-red light. We still expect both of those to pass, but you may need to do a little bit more work to satisfy yourself by looking in the genome browser. Perhaps you would need to add a custom candidate from an alternative allele that you think could be equally plausible, and compare the 2 alleles. And that is one way in which you could rule out anything else and therefore have confidence in the red and yellow call.
Thank you, Pete. There's one more question related to reporting, because in the presentation Derek has shown us, we've seen that TXT is an accepted format. So is it producible from Twin? Does it support the TXT export format?
So the HML format is fundamentally a text format. It's an open format. It does have HML tags, which is the histocompatibility markup language developed by the NMDP, but it is also fundamentally a text-based language. So if what you're asking is can you parse that information out of the text file and into something else, then yes, you will be able to do that easily. Can you load it with a text editor? Yes, you can. Does it include all of the consensus sequence information as text characters? Yes, it does that as well in one of the whole gene areas of the HML tags.
All right, thank you. Okay, next one. From what you've shown, it seems that most ambiguities arise because of the lack of completeness of the allele sequence. How do you propose we handle that in the short term?
I guess it's hard to tell from the question whether they mean incompleteness in the consensus sequence or incompleteness in the database. The reality is if you use the holotype method and are able to, where our focus is on recovering whole gene consensus sequences, you will almost never have any ambiguity unless we do not actually cover that area of of the gene, which in the DRB1 case, where the 1210 and the 1201, their exon 1 is not covered, so even though we have an unambiguous consensus sequence, we can't make an unambiguous typing. And that's the most common cause of ambiguity. The next most common cause of ambiguity is, of course, the cis-trans ambiguity with very long homozygous stretches between two alleles, which happens around 4% of the time in DPB.
Thank you. I think the next one is probably for Derek. Can you please give us a reference on how to submit or confirm rare alleles?
Hi, thank you. I think the best way is actually to go to AFND and submit the question to us, and then we will send you the program whereby it can interpret all your data at a quick flash to see if you have rare alleles and which ones there are and which ones qualify, and therefore then what we do with it. I think that's the easiest way to do it, if you don't mind to do that first. You can't submit any question to allele frequencies directly on the website for anything.
Thank you. I think there's one more for you. Is it possible to establish haplotypes frequencies differing between populations, countries, regions of the world, in a way that we could track populations, origins, migrations through the human history?
Yes, I think the answer is yes, and probably the haplotypes are better than the individual alleles to do that. At the moment, with this new submission of data, genotype data, we are collecting haplotype data which has been provided by the individual submitting, but I don't see why we can't move to actually getting the genotype data and calculating the haplotype frequencies ourselves. But at the moment, that's what's happening. People submit raw data, ambiguous, raw data, unambiguous, haplotype data, if they have it. And if they haven't, then I think the move is on for us to calculate those frequencies and have it. But certainly, I think you'll get a lot more information from haplotype data than you will from individual alleles.
Thanks, Derek. Next question. It's actually two questions in one. What is concordance? And do you have a benchmark work to compare with current or traditional SBT?
So concordance is, of course, defined by the two different technologies being compared. So you have to define that up front. So if you're asking what is the concordance between SBT and NGS, or what is the concordance between two NGS technologies, or you do need to define what concordance is being measured between. But to the question of do we have any data of SBT and holotype data, absolutely. We've actually published an ASHI in a couple of posters and in our symposium a lot of data. It's actually not just SBT data though. It is SBT, SSO, and SSP data in some cases. What we have observed is that at the two-field level, there is 99.95% accuracy of holotype for 5 and 7 loci, but the concordance can actually be slightly lower, and that's because we were able to determine upon reflexive testing that there were actually some errors with the sample, with the original typings that we compared against. This does take into account that we were using some registry samples which may not have been typed to the same accuracy as the clinical samples that were submitted to us, because we had a range, but the background error rate in the legacy technologies was around 2.5%, whereas the accuracy of holotype was 99.95%, so only 0.5% incorrect.
Okay, the next one. Is there a way for Omixon Twin to integrate the information from the allele frequencies database to give more confidence in the HLA genotyping when needed?
Yeah, I think so. We used to have an expression, and maybe Henry Ehrlich had first come up with this, is really to know your population. And it was one of the things that we did back in Belfast to do the alleles to two fields, whereby we did this in the first 1,000 on a bone marrow registry. And we were able to know our population and know if we only were able on certain occasions to define the one field what the estimation would be and what the likelihood, especially in a homogeneous population, would be to two field. So that in itself can give you encouragement. But I think that there is a lot that next generation sequencing and hopefully AF&D can work together to do this, especially I think the rare allele situation whereby, as Peter's been talking about, rare alleles, what we're trying to collect is that rare allele with a haplotype, and therefore that would give you greater confidence that you were actually detecting the particular rare allele, because if you know the rest of the genes on that haplotype. So I do think that two things go hand in hand, but the main point is to get the data. We, and I'm sure Omixon are the same, certainly in AF&D, we're only as good as the data that we're giving, and it has to be a two-way situation. People cannot expect the data to be there if they're not giving it. So I plead with all colleagues throughout the world to give us this data and give us this raw data, and from that, no matter who is using it, it will build up a great deal of confidence in what we're trying to do.
And a small technical comment to answer that question as well is that if it is in Derek's database, currently that is accessible only through the web as I understand it, but an offline version of his database could be integrated in a tool like Omixon to provide that data to the user.
And I think we have some more time maybe for one more question. There's one related to the kit. In your 11 loci kit, DRB345 are amplified in 3 PCR reactions or in 1? It's very technical, so
that's a great question. The structure of the kit is actually the DRB3 is co-amplified reliably with DRB1, and we disentangle those bioinformatically. DRB4 and DRB5 are separate reactions, and then they are pooled together along with all of the other loci, and then disentangled bioinformatically by the software after the sequencing run.
All right, thank you. I guess if there are any remaining questions, we will answer all of them by email. And Pete, do you want to wrap up?
Yes, thanks very much to Marcello for emceeing today. Thanks very much to Derek for coming. We're all going to head out and enjoy dinner in Budapest this evening together. But thank you to everyone who has signed in and listened to this webinar. We look forward to seeing you again soon.